Okay, so the third step in cellular respiration that we're going to be talking about is called the Krebs cycle. The Krebs cycle is also known as the citric acid cycle. So these terms will be kind of used interchangeably. So if you ever see citric acid cycle or Krebs cycle on a test or exam or a quiz, it just means the same thing. It's just another word. So the Krebs cycle was actually discovered by a man named Sir Hans Krebs in 1937. That's why it's called the Krebs cycle. Another important point that you need to understand is that this whole cycle that we're going to be drawing out is occurring in the mitochondrial matrix. So it occurs in mitochondrial matrix. So if we can recall, the mitochondrial matrix is inside of that inner membrane that we drew on the pyruvate oxidation note. It's inside of that inner mitochondrial membrane. So if we can recall from pyruvate oxidation, we were actually left with a molecule that was named acetyl coenzyme A. So that's what we left off with in pyruvate oxidation. So that's actually where we're going to start with the Krebs cycle. So we're going to write acetyl coenzyme A here. And during this whole cycle, instead of drawing out the molecules which we've been doing for glycolysis as well as pyruvate oxidation, we're just going to use these little circles to represent our carbons, just because there are a lot of molecules that are involved, and we don't want it to become too confusing for you guys. So we're going to start off with acetyl coenzyme A. So acetyl coenzyme A is actually a 2-carbon molecule. So there's our 2 carbons, and we're going to attach that coenzyme A here. So there's our acetyl coenzyme A. We're then going to draw a 4-carbon molecule here. 1, 2, 3, 4. This molecule we've actually seen before in C4 plants. It is called oxaloacetate. So that is a 4-carbon molecule. What happens is that an enzyme is going to come in, and it's going to actually attach this acetyl coenzyme A onto oxaloacetate. And what do we get? We get a 6-carbon molecule. This is called citrate, also known as citric acid. Which makes sense. That's why it's called the citric acid cycle, is because that is actually the first product that we see within the cycle. How is this occurring? Acetyl coenzyme A is attaching itself onto oxaloacetate, creating a 6-carbon molecule, which makes sense because we've got 2 carbons here, 4 carbons here, creating this 6-carbon molecule called citrate. It's important that we write which enzymes are creating these reactions and what are actually driving the reactions. So right here, we're going to write the enzyme that's responsible for attaching that acetyl coenzyme onto oxaloacetate to create that citrate, and that is called citrate synthetase. So again, citrate synthetase is going to grab the acetyl coenzyme A, it's going to attach it onto oxaloacetate, creating this 6-carbon molecule called citrate, or also known as citric acid. So what I'm going to do is I'm actually going to use this pink highlighter to just kind of draw this little cloud around all of the enzymes that we see throughout this cycle, just so you guys have a clear picture of where the enzymes are and what they are actually doing. So the next step, we're going to go in a circle, since this whole reaction is going to be a big circle. So we have citrate. Citrate then actually turns into an isomer. So if we can recall, an isomer is actually the same molecule. We're not taking away and we're not adding any atoms. We're just rearranging the shape of the molecule. And this is called isocitrate. So again, same amount of atoms. We're just kind of rearranging the shape, which we're not going to see here because we are just drawing it in a very simple way with the carbons. But just know that citrate turns into isocitrate, which is an isomer of citrate. So the next step, we're going to go in a little bit of a circle here. So the next step is that isocitrate is actually turned into a 5-carbon molecule. So we're taking off one of these carbons. So we're going to draw that here. 1, 2, 3, 4, 5. So there's a 5-carbon molecule. This molecule is called alpha ketoglutarate. So this is a new molecule that's created called alpha ketoglutarate. So we took off a carbon. So because we took off a carbon, that carbon's actually released as carbon dioxide, which I'm going to do in green. So that is now released as carbon dioxide. Another important thing that happens here is that when isocitrate is turning into alpha ketoglutarate, electrons are being lost because this is requiring electrons to be lost to turn isocitrate into alpha ketoglutarate. So what happens is that NAD, which we've seen before, NAD plus, is going to come in, it's going to pick up those electrons, and it's going to become its reduced version, which is NADH. Okay, so NADH just came in, it grabbed those electrons that are being lost from this reaction here, and it's going to turn into its reduced form called NADH. So this is now a high energy electron carrier, which is going to be needed in the next reaction, in the next steps of cellular respiration, which we will talk about after completing the Krebs cycle. So one last thing that's important that we need to write down is what enzyme drives this reaction. It is called isocitrate dehydrogenase. So isocitrate dehydrogenase. All right. Isocitrate dehydrogenase is driving the reaction of isocitrate turning into alpha ketoglutarate. That 5-carbon molecule, CO2, is released because we lost a carbon. NAD comes in, picks up those electrons that are being lost here, turns into NADH. So here's our enzyme. It's responsible for that. All right. So we're going to go into our next step here. So we're going to draw this way. We're going in a circle. So what happens is that alpha ketoglutarate is going to be turned into a 4-carbon molecule now. So a 4-carbon molecule, 1, 2, 3, 4. But what's different about this 4-carbon molecule is we actually are adding on a coenzyme. Okay, so we are actually going to add on this coenzyme group. So this coenzyme A group is going to come in. There's a coenzyme A coming in. It's going to attach itself onto this 4-carbon molecule. And again, what we see is that we went from 5 carbons to 4 carbons, thus that carbon's exiting as CO2. And one more thing that's important, again, we're losing electrons as alpha ketoglutarate is turning into this molecule, which we're going to name in a second. Thus, NAD comes in, NAD plus comes in, picks up those electrons, turns it into its reduced form, which is NADH. Thus, we have now created another highly energized electron carrier molecule. So let's name this guy first. So this guy is actually called succinyl coenzyme A. So succinyl coenzyme A is this molecule right here. And it is important to label the enzyme that drove this reaction. It is called alpha ketoglutarate dehydrogenase. Okay, So what we did here is we got our alpha ketoglutarate. We then turned it into this succinyl coenzyme A by losing a carbon, releases carbon dioxide, adding a coenzyme A group, and that NAD comes in, picks up those electrons, turns itself into NADH, right? So the next step, we're actually going to be heading up the circle now. So we're going to head up the circle, and we are going to actually lose this coenzyme A group. So we saw that coenzyme A group was added on. It is quickly going to detach itself again, creating a 4-carbon molecule. 1, 2, 3, 4. This is called succinate. So we're going to draw that coenzyme A leaving. So it's exiting. It is leaving. As you can see, it's no longer attached. And this is called succinate. So it's important to label the enzyme that's responsible for driving this reaction once again. And this is called, succinate coenzyme A synthetase. Okay, so there's the enzyme here, and something interesting happens at this point in time right here that we're going to talk about. So what we are going to be seeing here is that ADP is going to turn into ATP. So at this stage of this reaction, we're actually getting energy, but we actually need a helper. So we need this helper, and it's called GDP. So GDP comes into the reaction plus an inorganic phosphate. GDP plus an inorganic phosphate creates this molecule called GTP, very similar to ATP, but a little bit different. So this guy's kind of a helper that's going to help us create ATP. So what happens with GTP is that GTP is actually going to donate its phosphate group. So I just kind of did a dotted line here. It's going to donate its phosphate group to a molecule that we've seen before called ADP. Right, so ADP now has gotten that phosphate group, thus it's going to turn into ATP. So there's our ATP that we've seen. That's that energy. So this is just going to continue to happen. So again, GDP comes in with an inorganic phosphate, turns it into GTP. That GTP donates its phosphate group to ADP, which creates ATP, which is that energy. So just think of GDP as a helper within the Krebs cycle that's going to help us create ATP. Okay. So next, succinate is going to turn into a 4-carbon molecule. Once again, 1, 2, 3, 4. And this is called fumarate. So there's fumarate. What drives this reaction here is an enzyme called succinate dehydrogenase. So let's draw our little cloud around it so we know it's the enzyme that's responsible for this reaction. And we're going to actually see a new molecule come in. I'm going to use blue. So this is very similar to the NAD that we've been seeing. It's actually called FAD. So FAD is going to come in. FAD comes in, again, as I said, when these redox reactions are happening, we're losing electrons. So electrons are being lost during these reactions. So FAD comes in and it picks up those electrons and it reduces itself to FADH2. So it actually has protons as well. So FAD accepts the electrons and the protons, leading to FADH2. So this is now an electron carrier that's very similar, as I said, to NADH. And it is now carrying electrons as well as protons that are going to be used in the next stage of cellular respiration. Okay, so we're almost at the end here. So fumarate, we have here, is going to turn into a new molecule. 4 carbons, 1, 2, 3, 4. It is called malate. And how it actually turns into malate is a water molecule actually comes in. So H2O. So water, there's this input of water to create malate from fumarate. And what drives this reaction, we'll put here, is called fumarase. So there's our enzyme. And then lastly, the last thing that happens, malate turns back into oxaloacetate. Okay, so the way it does this is through an enzyme called malate dehydrogenase, which again, we'll do our pink cloud here. All right. When malate gets turned into oxaloacetate, again, those electrons are being lost. Who's picking it up? We're gonna have our friend back. NAD+ comes and picks those electrons up, which then gives its reduced form of NADH. Again, we've got another high energy electron carrier here, NADH. That's going to be used in the next step of cellular respiration. Right, so there is our Krebs cycle. So let's just do a quick little summary at the bottom here to kind of see what we have. So what are our final products? What did we end up with? Our final products. And remember, I should have mentioned this at the beginning, but everything is actually happening twice. So because we ended off pyruvate oxidation with the 2 acetyl coenzymes, right? So everything is actually going to be happening twice, because if you can recall in pyruvate oxidation, we actually created 2 molecules of acetyl coenzyme. Because at the end of glycolysis, we created 2 molecules of pyruvate. So this is all happening twice. So what did we get? So what final products did we get? Just gonna move this here. So the final products. So we've got 1 NADH here, so actually 2, 4, 6. So we created 6 NADHs, because remember it's happening twice and we see it 3 times, 1, 2, 3. So we've created 6. We have created here, we've got our ATP, so we've created 2 ATPs. And lastly, we have this new molecule, FADH2. So we've created 2 FADH2. Okay, so those are our final products that we have. Here's our whole note. So these final products are going to be sent to the next step of cellular respiration, which we're going to talk about, which is called oxidative phosphorylation. So these 6 NADHs, these 2 ATPs, and 2 FADHs that we created within the Krebs cycle happening in the mitochondrial matrix are going to be sent to the next step, which is called oxidative phosphorylation.